What if we were to tell you that a group of students is currently collaborating to create super bacteria? What if these bacteria could one day replace antibiotics? What if they were able to fight against any type of pathogen and could be programmed in a simple way according to our needs? Sounds crazy, right? And this technology could be the solution to major healthcare issues. Even crazier, right? Well, this is currently taking place. We are Team IONIS Paris, 10 last-year engineering students actively working on developing a tailor-made, innovative, super E. coli. Bacteria can be found everywhere. Most of them are not dangerous for us. In fact, more than half of our body is not human. It contains millions of bacteria which help us survive. But others can infect us and potentially kill us. Let's rewind back to a century ago. Once contracted, a bacterial infection would most likely lead to death, no matter your age. Picture yourself running to work, for example, tripping and hurting your knee. Today, this is just a treatable wound. But in the past, even a small cut could kill. Can you imagine living with this risk? Thankfully, a revolutionary compound was found. In 1928, Alexander Fleming discovered penicillin, the first antibiotic which could save millions of lives by preventing the growth of bacteria. Patients could now expect to live much more thanks to antibiotics. Nowadays, they are an essential part of our modern lives and sometimes tend to be taken for granted. We are currently coming to the end of the antibiotic era. What is approaching is the era our ancestors lived a century ago, where simple infections would most likely kill. Scary data is being published. Can you imagine that if a solution isn't found by 2050, these untreatable infections could lead to 10 million deaths every year? But how? How have we come to this desperate healthcare situation? Today, the powerful drugs, known as antibiotics, are sometimes thought of as merchandise. According to the World Health Organization, in some countries, more than half of the antibiotics are misused. Natural selection is favoring the evolution of resistant bacteria, as they have natural processes which encourage their resistance to antibiotics. Since bacteria have the capability of transferring this genetic material between each other, these genetic advantages are easily spread. Is this a shocker? Actually, no. Even back in the 20th century, Alexander Fleming warned society that bacterial resistance had the potential to defeat antibiotics. Sadly, he was right, because a few years later, in the 1940s, bacterial resistance started to appear. Take a look at this data. Penicillin resistance appeared 12 years after its discovery. Carbapenem resistance took 8 years, and tetracycline resistance took only 1 year to appear. Antibiotics are becoming inefficient. There is a lack of investment and innovation in the field. This is a major threat to global health. What if we could develop a way to fight against antibiotic-resistant bacteria and save lives? How do we ensure that it could have a positive impact for society? This is where the IONIS Paris team comes in with BacTail. Target, kill, and self-destroy. An innovative way to safely and locally fight against antimicrobial resistance. We are developing a tailor-made, engineered therapeutic probiotic. Since we are using E. coli as a host, it can be effectively controlled. 4 steps. 1. Specific recognition and binding to the pathogen thanks to certain proteins. 2. Secretion of specific antimicrobial peptides to kill the target. 3. Self-destruction of our bacteria once they have accomplished their mission. And 4. Delivering our probiotic to its site of action. Okay, so now that you are aware of our goals, let's get into the methodology. How are we going to be able to achieve these experiments in the lab?
Phase 1 consists in specifically recognizing and binding to the target bacterium. But how can this be achieved? This is where our team was inspired by the natural predators of bacteria, which are bacteriophages. Let me explain. One particular class of bacteriophages, namely the Myoviridae family, possesses little legs or docking points called long tail fibers. Thanks to those legs, bacteriophages are able to specifically recognize and bind to the target. The core idea of the BacTail project is to give the bacteria the ability to recognize other bacteria the same way bacteriophages do. Thus, our name, BacTail. For safety and security motives, the model organism for our proof of concept is Escherichia coli K12, which is called a chassis in synthetic biology. Now that we have a host bacterium, which should be its target? In nature, T4 bacteriophages are specific to E. coli. The most distal region, the gene product 37, GP37, confers the recognition and binding ability to the membrane of the pathogenic bacteria. Therefore, for phase 1, the proof of concept consisted in expressing the recognition site, region GP37 of the LTF, on the outer membrane of our model organism. So, we created the following biobrick to execute this task. The major component is the GP37, which confers the recognition ability. GP37 will be directed to the surface of our chassis on its outer membrane thanks to the co-expressed LPP-OMP-A gene. Between the LPP-OMP-A and the GP37 is a specific cleavage site for a particular enzyme called TEF protease. This cleavage site is followed by an His tag and a red fluorescence marker, RFP, allowing to localize the LTF in the chassis. And finally, the two chaperone proteins, GP38 and GP57A, are needed for the assembly and the proper folding of the GP37 receptor binding protein. On the top of all this lab work, we designed a software tool to ease the biobrick construction to target different pathogens. Thanks to existing literature, our team incorporated the specific recognition site of bacteriophages directly into the software. Try it yourself directly on our wiki website.
Now that our target has been approached, the next step is inducing its death. But how can this be achieved? We have decided to look into bacteriocins as our weapon to kill the target. So, what exactly are bacteriocins? They are a type of antimicrobial peptide. These are small proteins naturally produced by bacteria to survive in highly competitive polymicrobial environments. They are considered as non-harmful for humans, and some are already being used in the food industry. After research and talking to specialists, we found a type of bacteriocin called microcin PDI for proximity-dependent inhibition. It was initially identified in 2009 in an E. coli strain found in cattle. Interestingly, it requires physical proximity between the producer and susceptible strains. Its narrow spectrum allows it to target lab strains of E. coli as well as pathogenic ones and lead to their lethal membrane damage. This makes microcin PDI an ideal candidate for our proof of concept. Let's look into the gene sequences that code for this microcin. Microcin PDI is a cluster of 5 genes. MCPM codes for the secreted microcin. MCPI procures resistance to the microcin for the host. MCPA is required for activation of MCPM, and MCPB and D are required for the extracellular secretion. Natively, the expression of microcin PDI is controlled by osmotic pressure. For our proof of concept, we wanted the expression of microcin to be constitutive and independent of osmotic pressure. That is why we redesigned the cluster by keeping all 5 coding sequences and using promoters and ribosome binding sites found in the iGEM registry. We use the Salus Lab RBS calculator to predict the translation initiation rates of each start codon and have the same one as for the native cluster. All in all, our system should allow the host to express the microcin and constantly secrete it. The host is maintained near the target thanks to the LTF. This allows the toxic concentration of microcin to accumulate and induce the target's lethal membrane damage.
Great! Now our bacterium has accomplished its mission it is time for it to be eliminated via a kill switch. This is phase 3. A genetic tool will ensure the safety of our system by controlling the life cycle of our bacteria. But why is this so important? Uncertainty. This is the main problem linked to the release of transgenic bacteria. Bacterial interaction with the environment cannot be perfectly predicted. There are risks of unanticipated survival and reproduction, but also uncontrolled transfers of genetically engineered DNA to other organisms. Throughout our research on AMR, we got in touch with the team IONIS Paris, who was also working on this subject. But due to the COVID-19 pandemic, a major lockdown began in India and the access to the lab was restrained. IONIS Paris was also looking for a kill switch system for their bacteria. So what a great coincidence! Thus, we decided to establish a partnership to help them run their experiments. The conception of the biobricks was done by the team IONIS Paris. The inducible system was inspired by the L. arabinose operon. The main genes downstream to the pBAD promoter were replaced by the killing system. Death-inducing bpDNase 1 will hydrolyze the double-stranded DNA. And upstream to pBAD, but in the opposite direction, is the gene coding for the regulatory protein RSC. But how is this whole system regulated? The presence or absence of specific components will determine the expression of the genes. In absence of L. arabinose, the RSC dimer binds to the specific alpha sites, forming a DNA loop. This loop excludes the RNA polymerase from binding to pBAD. This prevents the transcription of DNase 1. On the contrary, when L. arabinose is present, it binds to RSC to induce conformational changes. RSC will then bind to another alpha site, giving access for RNA polymerase to bind pBAD. This leads to the production of bpDNase 1, which destroys our engineered bacteria.
The fourth and last step of our project is determining the galenic form and the conditions under which our engineered bacteria will be stored. We will use a probiotic because our final goal is to fight against intestinal infections. These are living microorganisms which, when administered in adequate amounts, confer a health benefit for the host. But wait! Look at all the obstacles: stomach acidity, destructive enzymes, high temperature, and lack of oxygen in the gastrointestinal tract. So, we wondered, how will our bacteria be able to survive these hurdles before reaching their target? Plant-based hard shell capsule, that is the solution. They are resistant to acids and enable a delayed release. Plus, they are approved by a larger public as they are not made of animal-based gelatin. Finally, concerning stability during processing and storage, we opted for lyophilization, aka freeze drying.
For phase 1, to prove the expression and the localization of the receptor binding protein, GP37, on the surface of our chassis, we came to target the co-expressed yeast tag. We used a monoclonal anti-yeast antibody conjugated to the FITC, which has excitation and emission spectrum peak wavelengths of approximately 497 nanometers and 519 nanometers. As depicted on this outline, if the GP37 is well directed to the membrane, the anti-yeast antibody will bind to the yeast tag. This binding emits a signal read by a fluorescent absorbance reader. The results of this experiment are difficult to interpret. The samples containing the yeast tag emitted a signal similar to the control which did not contain the yeast tag. We believe that the antibody must bind unspecifically, but due to lack of time, we could not repeat the assay, and future tests should be conducted.
For phase 2, we created a system to produce and secrete microcin PDI to lyse the target bacteria. Once the system assembled and inserted into the chassis, we evaluated the production of MCPM, the 8.2 kilodalton precursor protein, by conducting a purification using the histag followed by an SDS page and necromocy staining. As depicted on the gel, there is a possible presence of MCPM in the sample as a band is observed below 10 kilodalton. In parallel, to confirm the functionality and activity of MCPM, we performed an agar diffusion assay. The goal is to see if the microcin-producing engineered bacteria on the discs enable the lysis of the bacteria. The results are positive as an inhibition radius is observed, meaning target cell lysis occurs. All in all, the results for phase 2 are very optimistic and future replicates should be conducted.
For phase 3, thanks to our experiments, several clones supposed to present recombinant plasmids with either the MRFP or DNase 1 gene were obtained. These plasmids were then verified by 4 different validation assays: a colony PCR, a linearization of the plasmid, an insert release, and an additional double digestion. And among 19 clones that we picked, 3 were showing positive results for 3 verifications out of 4. Arabinose is supposed to activate the expression of the gene following the PBAD promoter. Thus, the expression of the DNase 1 gene for the interesting clones was studied in the presence of arabinose. There, we found that the OD measured with the most verified DNase 1 clone was reducing over time when arabinose was added. This suggested that BPDNase 1 was produced, leading to cell destruction. Unfortunately, we didn't have the time to proceed to further investigations on these clones, but according to those results, it seems very promising.
Regarding human practices, our goal was to talk about AMR and BacTail with a panel of experts. Considering their point of view helped us shape our project. Talking with scientists from the Pasteur Institute allowed us to assess the scientific reliability and feasibility of BacTail. Insight on the urgency of treatment after diagnosing a bacterial infection was given to us by an infectious disease specialist. Discussing with a research director at the French Agency for Food, Environmental and Health Safety informed us on the regulation perspectives of BacTail. We were also curious about the consequences of AMR on animals and livestock production, so we contacted veterinarians and breeders. Finally, to determine our project's wider impact on society, we contacted a philosopher specialized in science. Throughout the year, our aim was also to raise social awareness on AMR and synthetic biology. For our public engagement, we did video conferences and presented global scientific notions directly to the public, thanks to events like a science festival organized by the City of Science and Industry, 2 webinars, presentation, Kahoot quizzes, etc.
Safety has driven our project conception. Every action we took was thought to preserve the safety of the lab team, society, and the environment by thinking about the present experiments and the future impact of our project. All manipulations were safely performed in our Class 1 laboratory and were not intended to leave it. Furthermore, before entering the lab, everyone had to read a good practice guide for R&D projects, which are specific safety and security guidelines given by our laboratory principal. In the future, to ensure full safety, we carefully designed our bacteria with a self-deletion system that will be activated once the chassis is effective and has performed its mission.
Future perspectives are testing our system with other targets, meaning changing the LTF at the surface of our chassis as well as the secreted antimicrobial peptides. The different plasmid designs can be done thanks to our software tool. A limit regarding bacteriocins is that some resistance can occur when there is a continuous exposure. This issue can be solved by secreting different combinations of peptides. The partnership with the ISER therapeutic team allowed us to test a type of kill switch that could be useful for our future system. As this consists of a proof of concept, future optimization will be needed to adapt the kill switch to its use in the human body. The existing possibilities consist in changing the inducible promoter or using a repressor. In the future, we could use E. coli strain Niso as our model organism. It has been used as a probiotic and therapeutic agent for over a century. All in all, after future optimization, we truly believe in the power of BacTail to specifically fight against pathogens.